This is a paper for IGCSE Biology for February-March 2022, and I just want to have a quick walkthrough, and this paper is for 1 hour 15 minutes. So quickly, without wasting so much time, let's see how we can approach to answer this set of questions. Now, question number 1 is for you to state the name of the gas exchange surface in human. The point of gas exchange surface in human is the alveoli. Now, they say state 2 features of the gas exchange surface in humans. Now, these are just the adaptive features of the alveoli that makes it able to carry out this process of gas exchange effectively. And that is, it has a large surface area, another factor there is that it's well ventilated. Number 3, you can write it has large network of blood capillaries. And number 4, you find out that this gas exchange surface is moist. So that's it for that question. This next question is say B, 1b, figure 1.1 is a diagram of gas exchange surface in humans. Yes, this is the trachea, this is the bronchus. You have bronchi, then the alveoli. This is the lungs itself. This is the external intercostal muscle, the internal intercostal muscles, and the diaphragm. So what is this question about now? They said draw a label line on the letter X on figure 1.1 to identify the external intercostal muscle. So this, you just write X, that's the external intercostal muscle. This is the internal, the alveoli, bronchioles, then here the bronchus, then your trachea filled with cartilages. Then the next question is state the name of the tissue that form C-shaped structure in the wall of the trachea and state the function. That is the tissue there is cartilage. And the function is it help to keep the trachea open so it won't collapse. Trachea open and prevent collapse. So air will flow easily. That's it. Then page 3, I said describe the effect of the thorax of contraction of the diaphragm. Now you should answer this question from the aspect of inhalation and exhalation. Now once the diaphragm contracts, contraction of the diaphragm, you find out that first the volume of the chest will increase because the contraction of the diaphragm makes air to rush into the lungs. So first the volume of the lungs increases. So you find out that the thoracic cavity, so therefore the cavities will increase. Now, what this will lead to is, it will also lead to decrease. This will lead to decrease in pressure of the lungs. Decrease in pressure of the lungs. So what will now happen is that air will now moves into the lungs. That's all. Now, they said table 1.1 compares the composition of inspired and expired air. Obviously, it's nitrogen. This should be because air has 21% oxygen. Then this is carbon dioxide. And variable and saturated, that's water vapor. So, the next question is complete the table which we've done. That's 3 marks. 2 is for gas B. Gas B in this case is oxygen. For gas B and gas C, explain the difference in the percentage shown in table 1.1 between inspired and expired air. This is inspired, let me use IN, and expired air. So this is straightforward. So answering this question, I can say gas B has a higher percentage of oxygen, because we've been able to identify the gas as oxygen, have a higher percentage of oxygen of around, always use statistics, 21%, of 21% of inhaled air, while it has 16% of exhaled air. Now, what does this signify? Now, this indicates that This indicates that 5%, because 21 minus 16, this indicates that 5% is absorbed during gas exchange, while gas C has a lower percentage in inhaled carbon dioxide, which is CO2, of 0.04%, while the exhaled content is 4%. That's all. Now here question 2 is, say figure 2.1 is a photomicrograph showing the fertilization of one human egg cell. So we're looking at just one human egg cell, and we have a lot of sperm cells around it. So let's see what the question is. Now the question is said we should describe and explain. So there are two major keywords here which meant to describe and explain the adaptation of the cells shown in figure 2.1 that enable fertilization and the development of the embryo to occur. Now, I'll pick two. There are two cells, so I'll start with the first one. Let me say sperm cell. Now, it's a 6-mark question. So, what are the adaptation of sperm cell and their responsibility? Explain it, how they are responsible in fertilization. Now, I'll pick first. All sperm cells have a nuclei, and the function is it fuses with that of the egg cell. Egg cell nuclei during fertilization. The second aspect, the second point I'll bring in is, I can decide to call this one, it has nuclei. Two, sperm cell have acrosomes, and you can say this is an enzyme. This is an enzyme that dissolves the jelly coat. It dissolves the jelly coat of the egg cell so the nuclei can penetrate. So the nuclei can penetrate. The third point here is they all have, sperm cell have a tail. This help the cell to move through the cervix, to the oviduct, where fertilization takes place. The fourth point, I'm just talking about sperm cell here, then I'll look at egg cell, is that sperm cell also have high concentration of mitochondria. Now, these help to provide the energy. This help in respiration. This help in respiration to provide energy. To provide energy for movement of sperm cell. Then next, I'll bring in the egg cell. Now, the adaptation is that egg cell have jelly coat on the surface. And what is the function of this jelly coat is it hardens after fertilization. To stop sperm from entering. Then another major function is it has cytoplasm. And the cytoplasm acts as a food store. It's a food store that help to release energy which help in the process of cell division of the zygote. Okay. Now, this next question is, they said people can use artificial insemination, that's IA, or in vitro fertilization to increase their chances of becoming pregnant. Outline the process of artificial insemination. You should just write an outline of it. First, you find out that the female is given fertility drug to stimulate ovulation. So females will be given fertility drug to stimulate ovulation. Then next, you find out that semen is usually collected from male. And inserted into the cervix during during the time for ovulation. So this increases the chance of the woman getting pregnant. That's all. Now they say outline how the process in IVF, in vitro fertilization, differ from artificial insemination. This is also quite simple, where IVF occur outside in a petri dish. Occur outside in a petri dish, while also IVF require many embryo is formed. Many embryo is formed in IVF, while in artificial insemination, you find out that fertilization is internal because you insert the sperm into the woman. Now, describe the social implications of fertility treatment. One of the implications is that it is expensive. Another implication is it may not be successful and need to be repeated. It is not always successful. Another point, it leads to multiple birth. And need more care. So babies may be weak and die eventually if not done properly. Babies may be weak and die. Another point is babies may become deformed. Babies may be deformed. babies may be deformed, and these which eventually have social emotional concern on the parent. So the next question is question number 3. Okay, question number 3 is here. A scientist investigated the effect. So we're looking at the effect of temperature on the mass of leaf picked from tea plant. So here, once given a question like this, you should always identify your independent variable. And you know, independent variable is what you change in the course of the experiment. So in this case, since we are looking at the effect of temperature, this will be our independent variable. And since we want to measure the mass of leaf, that will be our dependent variable. Now, there's 3 samples of leaf were picked, and the mass of each sample of leaf was recorded, and each sample of leaf was kept at a temperature at different temperature. So different temperature, that is what we are changing, for 4 hours. So if they are kept for 4 hours, then 4 hours is a constant in this case, which makes it a fair test. Now, after 4 hours, the mass of each sample of leaves were measured and recorded again. The scientists then calculated the final mass as a percentage of the initial mass for each sample. Then this shows the result of the 4 different temperature and final mass. They say we should explain the result. This is simple. So you find out that the higher the temperature, the lower the final mass as a percentage to the initial mass. So the higher the temperature, sorry, I'll just abbreviate here. The higher the temperature, the lower the final mass as a percentage. So you copy everything here as a percentage of the initial mass. So the higher the temperature, the lower the final blah blah blah. Then so you find out that at lower temperature, look at this, at lower temperature, that's from 5 to approximately around 20. At lower temperature, what happened? There is a gradual decrease. So it's gradual here. Full stop. Both. At higher temperature, the decrease is faster. It's more steep. So here is steep. Right? This part here is gradual. But at higher temperature, the faster decrease of the final mass as a percentage of the initial mass. Now, you can now bring in statistics. You can say let's go back. You say this is around at 4 degrees Celsius. At 4 degrees Celsius, you find out that the final mass as a percentage to the initial mass is, you go back, you check it, 90%. Why? at 30 degrees Celsius. What is happening here? At 30 degrees Celsius, you can check the mass here is 80 degrees. So you can just pick anyone at random. It must not be as I'm doing it. But at 30 degrees Celsius is 8%. So I just described the graph, but remember the question says you should explain, and it's 5 mark. So I should give reasons why what I just described is happening. So you find out that the higher the temperature, the higher the rate of evaporation. The rate of evaporation from the spongy mesophyll cells of the leaf, and also the higher the diffusion of water vapor out of the stomata. That will give you a 5 mark. Now they say state one factor other than temperature that would affect the loss of mass from a leaf. So here it can be state one factor other than temperature. Here you can have humidity and wind speed. All these affect the rate of water loss from plant. The next question is figure 3.2 is a photomicrograph of a tissue that transports water and mineral ions, and that tissue that transports water and mineral ions, you should know, is the xylem tissue. So let's see what the question is. The question is, state the name of the tissue. Xylem. That's it. So now they said describe how the tissue is adapted for its function. First, it's made up of a dead cell. So if it's a dead cell, obviously there is no cytoplasm. If it's a dead cell also, there is no cytoplasm, there is no cell wall, no end walls, sorry, END, no end walls. So this will help to avoid resistance to water movement. Next, you find out that it has lignin. Which makes it waterproof. And gives it support. This will give you a 3 mark. However, you can also add things like it's hollow. You can see the space, hollow tube for lateral movement. For the lateral movement of water and mineral. Next question is, they say explain how mineral ions enter plants. So simple. This is through active transport. through active transport, and it is usually what? Against concentration gradient. Against concentration gradient, using protein carriers and also it uses energy from respiration. That's all. Now, question 4. They said figure 4.1 is a diagram of Vibrio cholerae, which is the bacteria that causes cholera. So they give us the diagram of, so this is now a bacteria. So what is the question now? They said describe 2 similarities and 2 differences between the palisade mesophyll cell. Now a palisade mesophyll cell is a plant cell. So this is a plant cell. So what they are asking is we should give a similarities, 2 similarities and 2 differences between a plant cell and this bacteria. So if you look at the diagram, so both of them have ribosomes, that's similarities. Both cells have cytoplasm. Both plant cell and both cell have cell wall. Both cell have DNA. Yeah. So you can come, both have cell wall. Both have cytoplasm. Both have ribosomes. Sorry. Both have cell membrane. So the differences, you find out that these have plasmid. There is presence of plasmid. There is presence of flagellum, which is not found in palisade cell. But you can come and say, I can say palisade cell, which is a plant cell, have chloroplast. Another one is bacteria has plasmid. It has flagellum. Also, palisade cell have mitochondria. Why this doesn't? So, I think that is cool. Now they say explain how the cholera bacterium causes diarrhea. Now, how does cholera bacterium causes diarrhea? This is quite straightforward, but I found out that students normally have issues with this question, but it's a very simple question if you just do the steps that are involved. Now, first, you find out that bacteria will first attach. We attach to the wall of the small intestine. Now, once it does that, it will now release, the bacteria will now release a toxin that causes we release a toxin that causes the releasing of chloride ions in the lumen of the small intestine. Now, once that is done, this will decrease the water potential of the lumen. Decrease the water potential of the lumen. And once there is decrease in water potential, then you find out that lastly, water will now diffuse. Water will diffuse by osmosis from from the blood into the lumen, thereby causing watery physis. Causing watery physis. That's all that causes diarrhea, which can be treated. This diarrhea can be treated through oral rehydration, oral rehydration therapy, where you mix salt and water. Now, this next question is, they said, a scientist tested the resistance of one strain of bacteria to different antibiotics. And so the scientist tested the solution of 5 different antibiotics, A to E. She soaked a paper disc in each antibiotic solution. The paper discs with antibiotics were placed in a petri dish containing bacteria on agar jelly. Now, figure 4.2 is a diagram of the appearance of the petri dish after 48 hours. Now, the shaded area show where bacteria grow, while the clear area show where bacteria did not grow. So all this shaded part is where bacteria grow. So this clear part, bacteria did not grow, while the shaded area is where bacteria grow. And this is the paper discs with antibiotics. So the first question here is, the strain of bacteria used in this investigation causes a disease. Now, using the information in figure 4.2, explain why antibiotic E would be the most effective at treating this disease. So if you come back, antibiotics E have the largest area with no growth in antibiotics. So you just write it has the largest clear area with no growth of bacteria. That's all, 1 mark. Now, they said the result in figure 4.2 show that this strain of bacteria is resistant to antibiotic A. Now, 5 years ago, a similar investigation found that the clear area for antibiotic A was the same size as antibiotic B in figure 4.2. Explain how bacteria become resistant. Now, already you know that bacteria become resistant through mutation. How they become resistant? By mutation. And the next thing you should do here is define what mutation is. By mutation, through the changing in the base sequence, of the DNA. Changes in the base sequence of the DNA. Once the base sequence of the DNA change, it will now do what? It will producing a mutated allele. And you should remember that an allele is an alternative form of a gene. So a different form of a gene is now formed, producing a mutated allele that will make the bacteria survive when you apply antibiotics. That will make the bacteria survive and reproduce. Passing the allele to to its offsprings. So while the non-resistant bacteria will die out from antibiotics, non-resistant bacteria will die out, leaving a population of resistant bacteria. That's all. Now they said describe how to minimize the risk of antibiotic B developing the same result as antibiotics A. How do you resist the risk? Minimize the risk. How will you make antibiotics B not to become resistant? The first thing is complete dose of antibiotics. So you should always complete the dose given by the doctor. Next, you should take antibiotics only when required. Antibiotics only when required. And do not take antibiotics without doctor prescription. That's it. You just need one, but I'm just giving series, so anyone you remember, it's actually good for you. Do not take antibiotics without doctor prescription. That's it. Now, question 5 said the Arabian oryx and the northern white rhinoceros are both mammals. Now, figure 5.1 is a photograph of the Arabian oryx. Figure 5.2 is a photograph of the white rhinoceros. So this is the Arabian oryx, okay? This is a white rhinoceros. The first question is describe two pieces of evidence visible in figure 5.1 and 5.2 that shows that these animals are mammals. So if you come to the diagram, so you use the diagram to coincide. Both this has an external ear, and if you look closely, you find out that the skin has fur on it. It has hair, and this also have ear. It has hair. So the two things I'll just write here is the presence of external ears, and the skin has hair, or you can just put fur. That's all. Now the next part of this question state that different conservation methods are used to try to prevent species from becoming extinct. Now a population of the Arabian oryx and a population of the northern white rhinoceros were monitored. Now figure 5.3 shows how the population size of each species has changed over time. So you look at, always use your key because we are looking at two species here. So this one here represents the Arabian fox. Sorry, I'm saying fox oryx, and this other one is the rhinoceros, the white rhinoceros. Now, what is our first question? Is they say calculate the percentage increase in the number of Arabian oryx between 1990 and 2000. So you should know that this, if you answer a 3 significant figure, to calculate percentage increase is the final minus initial divide by initial times 100. So if that is the case, Using the year here, you find out that the final is will be 2000, the value for 2000 minus initial, which is the value for 1990, divide by the value for 1990 times 100. So what is the value for 2000? Let's go back and check. So sorry, for the year 1990, for the Arabian folks, which is this line, you find out that is this value here. So you trace it back to this, and this is 50. So in between here is 50. So you have if this is 50 and you have 10 here, so each there will represent 5. So if you look at that, this should be 50, this will be 60, and this will be 70. So this is around 70, while this is more somewhere here. And for which year? 2000 is this, and for 2000 somewhere here, if you trace it, which indicate around 260. So you just come 70, so 2000 is 260 minus 70, divide by 70 times 100. I'm not with my calculator, but if you do that, you should be able to convert your answer to 3 significant figure and write it down. Now, they say describe the data for the northern white rhinoceros shown. So they just want us to talk about the data then. So we have to use this to describe it. White rhinoceros. So if you look at this, you find out that the white rhinoceros started in 1980. So it started in 1980 and it increases and reaches its peak. So this is the peak. In which year? I think it reaches its peak in 2003. If you trace it down. So the peak is in 2003, but it started, so it appear in 1980, it reaches its peak in 2003 and decreases to I think 4, decreases to okay, this 50, this should be around this is 50, this is 10, 2 will give me 10, and that's 4 here, so that's to approximately 20. Okay, sorry, I'm using Arabian fox, I'm supposed to use rhinoceros here. So to approximately 5, if this is 5, this will be 1, 2, to approximately 2 in 2020. So that will give you a 3 mark if it's a 3 mark question. So that's it. So first you just write it appear in which year? 1980, based on the graph, and increases to its peak in 2003. So let's see the value. 2003, the value of approximately this 35, approximately 32, to 32, and then decreases to I think 2 in 2020. So you can add other values, other year, just choose to get your full mark. Now they say suggest the conservation method that were used to increase the number of Arabian oryx between. So yeah, what are the method of conserving species? That's all they are asking you. So first, you can, in this case for animals, you can have captive breeding. You can have gene banks, captive breeding. You can put zoo too. You can also have create awareness. You can also protect the habitat. Another thing you can do here is, okay, hunting. So you can have legislation against hunting. Against hunting. You can also have construct national park, construct natural parks and game reserve, and game reserve. Game reserves. Now, the next, I said, explain the risk to the northern white rhinoceros species as a result of its population size. If you look at the population size at 2000, that decreases to approximately 2 based on the graph, and the risk will be that there will be risk of extinction, and if it gets extinct, it will decrease biodiversity. We use multiply effect now, biodiversity, and that can also lead to increased competition. It will also affect the food chain. Decrease evolution. That's an effect. That is why it's dying out. Decrease evolution, therefore it cannot adapt to environmental changes. Now the last question, question 6. We are almost done. Complete table 6.1 to show the names, function, and site of the 3 different digestive enzymes. So pepsin, site of pepsin is stomach. Trypsin, the site of trypsin is the small intestine, the upper part, which is the duodenum, but you can just write small intestine. And function of pepsin is it breaks protein to amino acid. Trypsin is also, it has the same function, so it also breaks protein to amino acid. Now they say break down maltose to glucose. Maltose to glucose, the enzyme is maltase, and it takes place to the site is the small intestine. That's it. 3 marks. Now the next question is, enzymes are proteins. Yes, we know that. And they say figure 6.1 show the stages involved in protein synthesis. So the stages involved in the making of protein. And first, if you look at this stage 1, code for a particular protein, mRNA stage 1. So what will happen in stage 1 is the process of transcription, where the DNA will make a code to manufacture a particular protein in the form of mRNA. So transcription is taking place here. You should know that. Then let's see, B is that the code for the particular protein, look at it, which is now in the form of mRNA, is now moving out of the nuclear membrane, out of the nucleus. And if it moves out of the nucleus, it goes into where the cell is still inside the cell. Out of the nucleus, it now goes into the cytoplasm. So the mRNA will now move to the cytoplasm. And what will now happen in the cytoplasm is that the ribosomes will now, it will pass through ribosomes, and ribosomes will now translate. So translation will now take place here. So the first question, let's see the first question. It says, state the name of the part represented by the letter A and C. Let's see, letter A is transcription. transcription in the form of mRNA, while C here is passing through the ribosomes, so to make it, so in that case is name of the part, that would be the ribosomes. It's passing through the ribosomes, and what is happening there is translation. Describe the events that occur during stage 2. Stage 2 is when the mRNA, the code for the making a particular protein in the form of mRNA, is moving out of the nucleus into the cytoplasm. So you find out that what is happening is that the DNA transcribed into mRNA is in the nucleus. That's what happens in A. In the nucleus, what is happening in the nucleus, full stop. So the mRNA is transferred out of the nucleus into the cytoplasm. Out of the nucleus into the cytoplasm. Now, stage 4 determine the order in which the part labeled B is assembled. The order in which part labeled B is assembled, the order is through base sequence of the DNA or the gene. So that would be as a result of the base sequence of the gene on what? The base sequence of the gene that is copied by the mRNA. That's all. Then the last question is, the shape of a protein is very important to its function. The shape of a protein is very important to its function. So explain the importance of shape for the function of an enzyme. So simple. We're looking at as an enzyme. So enzyme have specific shape of active site. That's all you need to write. First part. That's the first thing. Enzyme has specific shape of active site. Specific shape of active site that allows the complementary substrate, because it's 3 mark. Complementary substrate to bind to form enzyme substrate complex. This will give you a 3 mark. Okay. So thank you, and please remember to subscribe.